The next presentation is an approach to bacterial conjugation through agent-based modeling in a predator-prey relationship by Londoño, Larrea, Juan Contreras, Universidad Central de Ecuador.
Okay, good afternoon. My name is Pablo Londoño Larrea. The work that I'm going to present you is an approach to bacterial conjugation through agent-based modeling in a predator-prey relationship. This is a work between a collaboration between the university, the Central University of Ecuador, and the Oak Ridge National Laboratory. So to get in context, what is a bacterial conjugation? A bacterial conjugation is a mechanism of DNA transfer between bacteria via cell-to-cell direct contact. So in this case, we have a donor and a recipient. The donor has a plasmid that is a small section of DNA that can be transferred to another bacteria, and the recipient receives that plasmid, and that small section of DNA get attached in the DNA of the recipient, and that has information about antibiotic resistance, ability to use new metabolites, and in general, adaptation to the environment. So this mechanism is very useful for bacteria to survive. So we understand this like a predator-prey relationship analogy. So in this case, we have a donor that is going to act like a predator and a recipient that is going to act like a prey. So in this case, we have this picture of an eagle trying to get in contact with the rabbit, you know? To model this, to try to simulate this behavior, we have this agent-based modeling that is a type of modeling where we have agents, agent-to-agent interactions, and also agent-to-environment interactions. In these interactions, we have behavior rules. So these behavior rules can be based on thermodynamics, chemistry reactions, biology, and other kind of behavior rules. So we use this NetLogo. NetLogo is a software, is a free software that allows us to code, to write down the rules, and to try to simulate, to have a model to simulate that kind of interactions. The last paper that we published is the application of the machine learning based on this model for Pantoia. Pantoia is a bacteria. So in this case, we can see the real and the simulation pictures. As we can see, is really similar. So using this information, we have a very successful representation or modeling of the growth rate of Pantoia in an agar plate. The Acton model has a problem statement on how to simulate the interaction of Escherichia coli as a donor and Pantoia species as a receptor. And the main application is because in the Oak Ridge National Laboratory we have a genetic modified Escherichia coli that has a plasmid that has a CRISPR-Cas9 type of plasmid that can kill Pantoia. This is really important because Pantoia is a very bad bacteria for our gut. It causes fever, diarrhea, and something like that. And also, this kind of model is useful because it reduces research costs via in silico testing instead of using the normal testing, laboratory testing, you know. So, in this case, I have this video of our model. As you can see, the green dots are E. coli, and the Pantoia are the red dots. So you can see how the bacteria is killing to the other bacteria. Yeah? We have 13 parameters, including aggressiveness, growth rate, and kill range. That is really useful for other bacteria to to simulate this kind of interaction. And also we have 3 test outputs: the growth rate, comparison between the obtained and the experimental, and also a fitness function. Something interesting of this is also that the optimization of this was done using genetic algorithms and machine learning techniques in a partial section of the supercomputer that we have in the Oak Ridge National Laboratory, but the optimized model can run easy in an Intel Core i3 PC. It's a normal laptop that we have in our home. So the conclusions are that we have successfully reproduced the interaction between Pantoia and E. coli compared with the real experiments. This model can be used as a template to simulate all the bacteria strains that we have in the laboratory, and also this tool reduces time and initial investment in biotechnological research via in silico testing. If you want to collaborate, or if you are interested in our work, you can contact us, Pablo Londoño Larrea, Pablo Araujo Granda, or Miguel Fuentes Cabrera. Thank you so much.